Is your DNA replication knowledge slightly lagging? Well, don't worry, I'm here to help. My name's Mr. White, I'm a teacher with more than 20 years experience. So the first thing to mention about DNA replication is that this always happens prior to cell division. And the reason for that is because, as we know from earlier topics, each new cell that is made has to have the same copy of DNA. So we need to duplicate or replicate this DNA prior to cell division. Now, DNA replication happens in several locations along the DNA strand, and the reason for that is because DNA is just so long, 3 billion base pairs long, and if we started at one end and replicated DNA slowly from one end to the other, it would take far too long. So this process that we're going to be learning about today takes place at several locations all the way along the DNA strand. Now, there are two key enzymes involved in this process that you guys need to know about for your exam. The first one is DNA polymerase. Okay, and we'll talk about the role of DNA polymerase in a second. The second one is ligase, and again we'll talk about the important role that ligase plays in replication in just a moment. But let's talk a little bit about DNA and how DNA fundamentally works. Now, we know that DNA from the structure video has two strands. Okay, it's a double-stranded helix. Now we've not had it in helix shape for this diagram just to make it easier to see everything. But what we can see is we can see the complementary base pairs, A with T and G with C. We can see the 5' and 3' ends of this strand and the 3' and 5' end of that strand. And as we know from the structure video, those are anti-parallel to each other. And that's important for us to remember so that we can understand how replication works. Now, when DNA replication begins, the two strands have to be unwound, they have to be separated from each other. Okay, and those strands are separated by breaking those hydrogen bonds. Okay, these hydrogen bonds that form between the nucleotide bases, those get separated, those get broken, which means that the DNA double helix can unwind, the strands can unwind. Okay, on one side, on one side of the original strand, we call that the template strand. On one side of the template strand, we are going to add a primer. Now, we add that primer to the 3' end of the template strand. Okay, so primers are added to the 3' end of the template strand. Now, what is a primer, you're asking? Well, a primer is simply a short strand of nucleotides which are complementary to a specific sequence on our template strand. So if that sequence on our template strand here is T, G and A, then the primer that goes with that, that's complementary with that, has got A, C and T on it. Okay, so a primer is a short strand of nucleotides which are complementary to a specific base sequence on the original DNA. Now, that primer always joins to the 3' end of the template strand. Okay, that's very important to remember. Now, on this side, on this strand, replication is fairly straightforward. It is what we call continuous. Okay, replication occurs continuously all the way along this strand, and the reason for that is because DNA polymerase adds nucleotides to the deoxyribose or 3' end of the new DNA strand which is forming. Now, this is the new DNA strand which is forming. Okay, now we know that because strands are anti-parallel, if that's the 3' end of the template strand, that's going to be the 5' end of the new forming DNA. Okay, and so therefore the 3' end of this new strand is actually up here. And so DNA polymerase will add DNA nucleotides to this end of this forming strand. Okay, so therefore it will bring in, what nucleotide will it bring in? It will bring in the nucleotide base G, because G is complementary to C. Okay, so we would bring in G here, and we do that because that's complementary to C. Okay, then new hydrogen bonds will form between it, and it will start to form this new strand here. Okay, so we can see that strand forming up here, and we've got G there. Okay, similarly it will go on and we'll get another base there. What's complementary to T is A. Okay, so the A will form there. Alright, now the good thing about the leading strand is this will just continuously go up like this. Okay, it will continue to add those DNA nucleotides to this end of the strand, and because this, what we call this the replication fork, that's the point at which the strands are unwinding, that will continue to unwind in that direction, which means that this bit will open up, this bit will open up, this bit will open up, and we can continue just to replicate that new strand of DNA, okay? So that is what's called the leading strand, and that replicates continuously, okay? Primer at this end, DNA polymerase adds the nucleotides to the 3' end of this new forming strand, the deoxyribose end. We've got G, then we've got A, and if we were to continue up, we'd have a T, then a C, then an A, and so on, okay? So it would use complementary base pairing to continue to form that new strand. Now, unfortunately, it's not as easy as that on the opposite strand. Now, this opposite strand over here, we call this the lagging strand, and we call it the lagging strand because it's just not quite as smooth or continuous in terms of replication over here. If we have a look, now if you've got to imagine what's previously happened here, which is that this DNA strand will have unwound down here at the very start. Now when it initially unwound, when these first few bases were exposed, we got the primer going in here on the lagging strand, no problem, but the primer that joined on the lagging strand, that one there can't continue to add bases in this direction, okay? This would be the 3' end of this new strand here, because that's the 5' end of the opposing strand. So therefore we can't add nucleotides in that direction. We need to add nucleotides in that direction. And this is why it's called the lagging strand, because what we then have to do is we have a primer here, but we can't form new nucleotides there. We then have to have another primer up here so that it can then add nucleotides down in this direction here, okay? And so we could add the C there, okay? So we add the C to that one there, and it goes in as this arrow shows, it goes in this direction here. So we're adding that G, that C to that G there, okay? And that would continue to form the strand. All right, so it's slightly trickier for the lagging strand. It has to replicate in fragments instead. Now those fragments eventually, so this fragment here, the fragments that are made up here and then up here and so on, those fragments will eventually be joined by an enzyme called ligase. Okay, it joins these fragments together on the lagging strand. Okay, so DNA replication, it's an important process. It has to happen prior to cell division. It happens in several locations along the DNA, and it involves replication continuously on the leading strand. It involves replication in fragments on the lagging strand, and those fragments are joined together by ligase. How do we actually make the new strand? Well, we add DNA nucleotides to the 3' end of the new DNA strand that's forming, and the enzyme that carries that out is our DNA polymerase. Okay folks, I hope that was useful. I hope that's covered the main things that you need to know for DNA replication in your exam. Thanks for joining me. I'll see you in the next one. Class dismissed.